Zinian. That was lovely to see some of that data coming out as well, especially. So next up we have Ollie Gray. So Ollie Gray carried out his undergraduate at Cardiff in the UK, followed by a PhD in Manchester, and then left academia, sadly, to go into the UK Biobank, and is now a senior data analyst there. But we keep getting him involved in things, so he's an honorary academic. Ollie is involved in supporting and developing the UK Biobank imaging project, where he's been involved in detecting acquisition issues, data provision to the research community, and especially recently working on tools for analysis on the UK Biobank's research analysis platform. So very glad to have Ollie here representing the Biobank.
Hello, thanks for having me. As Neil said, I'm here to talk about UK Biobank, which is a big open data set with global challenges and enormous promise. So what is it? For those of you who don't know, in 2006 through 2010, we had 500,000 volunteers to take part in a large-scale epidemiological study. They agreed to share their health-related data for general research, health-related research. UK Biobank participants have visited assessment centres up to 4 times and contributed further data through online questionnaires and follow-up studies. And this has led to a richly characterised cohort with available data describing lifestyle, environmental and biological features of each participant. Critically, UK Biobank has also linked data from the UK's National Health Service, and alongside the near 20-year duration of the study, this linkage has revealed a rich diversity of health outcomes and intermediary phenotypes. Importantly, data from UK Biobank are readily available for any type of health-related research by academic and commercial researchers worldwide. The value of UK Biobank is increasing with time. As the population of participants gets older, there are more incident cases of disease occurring in the cohort. Here you can see some predictions of the number of incident cases that will occur in the cohort over the next 10 years or so. We're expecting to see particularly large increases in the diseases of old age, like dementia. Participant visits to assessment centres have provided the opportunity to carefully characterise our participants. We provide them with tests of sensory and cognitive functions. We also measure their physical features and the operation of their heart and lungs. In addition to assessments made at the clinic, we also have provided a subset of the cohort with wrist-worn accelerometers and cardiac monitors to acquire data that is otherwise difficult to access. Online questionnaires, which participants answer at home, provide access to participants who can't visit assessment centres anymore and supplement assessment measures to further characterise the cohort. Participants also provide biological samples in the form of blood, plasma and urine at assessment centre visits, and the UK Biobank has provided these samples to research groups and organisations around the world that have derived data to describe participants at multiple scales. These range from the recently released whole genome sequencing data to the identification of biomarkers of infectious disease. I've highlighted some data sets here that cover all 500,000 members of the cohort, and further work is ongoing to expand coverage of the other biological assays and further biological assays to the entire cohort. With respect to imaging, the imaging study aims to provide some assessment of the whole body. I've just brought up the MR sequences that we have participants do here, but we also have DEXA and OCT images, and some ultrasound as well, and some ECG. So images from each target are provided in dicol format with virtually no modifications from the scanners, and all the technical specifications are available in the metadata from Biobank. All the brain acquisitions are done at 3T, and everything else is done at 1.5T. All the participant brain images are processed using the UK Biobank brain imaging pipeline. This pipeline was developed by Fidel and Steve Smith at Oxford, and a consistent set of structural, functional, and diffusion-focused analyses are performed on every single image. These primarily rely on established mechanisms within FSL and FreeSurfer, and recent expansions to this data, to this pipeline have included the quantitative susceptibility mapping and the arterial spin labeling derivations. The UK Biobank brain imaging pipeline and a Docker file implementation of the pipeline are freely available, and support continues to be provided by the FIMRIB group and UK Biobank for any researchers trying to utilize the pipeline or variants of the pipeline for analysis of UK Biobank data or their own data. The pipeline derives phenotypic characterizations of the brain. These numeric features tally to over 2,000 numeric features, depending on how you look at it, potentially many more than that. In the 2018 paper that describes the pipeline, the group performed univariate correlations with all the other tabular phenotypic data, which is shown here. And the majority of further studies that utilize the brain imaging at UK Biobank have used these data. In the field of epidemiology, many studies using UK Biobank utilize the very generic image-derived phenotypes like brain volume and gray matter volume, simple stuff across the brain. This is fine, and there's a lot of value in this, but there's also explored opportunities for collaboration between imaging groups and epidemiologists as the cohort grows and the incident cases grow, to really provide even more insightful findings. So reach out to your local epidemiologist and help them reach out to you. So in addition to the tabular phenotypes I just described, Biobank also provides intermediary files from the brain imaging pipeline for all modalities. One can pick up images, for example, that have already been aligned and tissue segmented, and these files provide a platform from which further analyses can be performed to your own specifications without having to start right back from the scanners, the images that come from the pipeline. In addition to these intermediary files, the pipeline produces and we provide image files and masks like the white matter hyperintensity mask shown here that provide a spatial aspect to some of the numeric data that is produced by the pipeline. Other research groups have also contributed significantly to the brain imaging space. Measures of sulcal anatomy were described in this contribution by a group at Biogen. After using FreeSurfer and the morphologist pipeline within BrainVisor to derive sulcal measures, the group utilized the UK Biobank genetic data to look for folding associations with genetics. The group then assessed these with a genome-wide association catalog to link to health and disease statuses. The UK Biobank has now doubled the number of participants with imaging data since this analysis was performed. And as I mentioned, the increasing incidence of health-related disease in the resource really provides opportunities for groups to collaborate across kind of distinct fields. This figure shows the kind of two-dimensional feature space, just the first couple of dimensions of all the image-derived phenotypes provided by UK Biobank. And I quite like this figure because it illustrates the kind of power of UK Biobank to describe the human brain, but it also demonstrates areas of sparsity which have yet to be explored. So potentially that's an area where some further work will kind of fill out this space, hopefully. This was the area that the Biogen group, the sulcal measures that they produced this kind of gap in, was filled by them. I described earlier the power that comes from the longitudinal nature of UK Biobank, and here I'm showing results from the UK Biobank brain imaging study that was performed during the COVID-19 pandemic. Here, participants that had already received brain scans prior to the pandemic were invited back during the pandemic, And this reinvitation targeted individuals that had suffered from COVID and some age-matched control participants. So this analysis, again from the group in Oxford, performed in this paper, assessed longitudinal differences between the pre and the post-COVID scans to identify regions that changed in association with disease. The authors identified changes in limbic regions associated with the olfactory system, and they hypothesized about neuroinflammatory events from COVID, loss of sensory smell, of input due to anosmia and lack of smell. So UK Biobank and the research community continue to expand collaboratively on characterization of the participants to provide the best opportunity to identify the causes, preventions, and treatments for human disease. However, it turns out that human beings are quite complex, and the data that captures this complexity can take up quite a lot of space. The UK Biobank genetic data is colossal and makes up the vast majority of the UK Biobank's storage requirements. However, we still have 350 terabytes of imaging data, and we shouldn't sniff at that. And reasonable attempts to analyze this data require specialized storage and research computing. One increasing feature of UK Biobank is the international attraction of the resource, hence my presence here. In 2016, just over half the UK Biobank researchers were from outside the UK. And in 2021, this stood at 80%. I think has still continued to increase since then. This raises the important question of how to democratize access to the data when many researchers do not have access to the resources required to effectively analyze the data or even to store the data. A research group shouldn't be forced to build a data center or lose agency over their research in order to realize the full potential of UK Biobank. The UK Biobank Research Analysis Platform provides access to as much of the UK Biobank data as you want, whenever you want it. This data is provided in fairly intuitive structures which can be accessed as on demand. The platform also provides access to a plethora of AWS compute instances that you can tailor, so that you can tailor your compute to your use case. And importantly, we also consider tooling, as Neil said, as a high priority. So the UK Biobank is an amazing resource for epidemiological research, and the UK BRAP provides tools to enable this. So there's a cohort browser which provides mechanisms to allow filtering of the cohort and produces simple but effective visualizations that can be readily exported for summary level figures. In addition, databasing in Apache Spark simplifies and empowers access mechanisms to all the tabular data and brings the tabular data from the assessment centers and the questionnaire data into a common format and access mechanism with the health-related data from the hospitals and things like that. So the UK BRAP provides RStudio and Stata interfaces to further make epidemiologists feel at home. And lastly, importantly, you can bring your own data onto the RAP. So within your own legal and kind of frameworks, this allows you to compare across cohorts and further accelerate your research. The UK BRAP caters to both image analysis development and, critically for big datasets, analysis deployment. So the platform provides Linux-based instances and root permissions so that you can install whatever you need. You don't need to wait for research IT to install something for you anymore. The RAP also integrates snapshots and session management so that you don't have to start from scratch every time, and you can make sure that you never lose any of your work. And it also provides JupyterLab and MATLAB implementations so that creating visualizations is easy. And lastly, the platform has pre-installed versions of FSL and FreeSurf, so you can get working straight away, especially if you're a student and it takes a while to get these things set up. In deployment, you can take advantage of machine learning and GPU instances, and you can also take advantage of containerization and workflow languages to build your own tools for optimal throughput. It can be very, very cheap to run analyses on the RAP. It takes a bit of getting used to, but yeah. UK Biobank has recently launched the Global Research Access Fund. This aims to provide researchers at institutes in low and middle income countries with the funds to make the most of the entire UK Biobank dataset. If you're eligible or you know someone who is, then please do get in contact. I can always provide that email address again later on. The UK Biobank Platform Credits Program is also available to researchers from these low and middle income countries, but is also available to early career researchers. The £1,000 in Getting Started credits is available to allow you to explore the platform, get to know it in detail, develop and test tools and methods, and undertake analysis to support your research project. Further requirements for your research, like trying to run the whole cohort through a pipeline, thereafter covered by the enhanced credit. If you want to apply or you want further details, there's a QR code there for you, but very useful for your students. Where can you go if you need assistance? Well, DNA Nexus and UKB collaborate to provide documentation on the RAP and how to use it. This includes instructions for onboarding to the all the way through to tool building and deployment. And we're actively developing this at the moment, so we'll hopefully be improving in the near future as well. And then UKB and DNA Nexus also have their own GitHub accounts, which provide notebooks for accessing data, doing simple analyses. Again, we're also actively developing this, and any ideas for further guidance that you'd like to see or things that you're struggling with that we could give to other researchers here, be very, very welcome. DNA Nexus have a YouTube channel. They give some instructional videos there on how to use it. We also have a drop-in on Thursday at the conference room E4. And this is for UK Biobank researchers. If you haven't used the RAP yet, or if you're kind of just starting to explore that space, please come along, or if you know somebody who does. And we're also interested at that drop-in in getting feedback from you. So is there things that are working well? Are there things that are barriers to you? And do you have any unmet needs, basically? Okay, lastly, thanks to all of our funders and especially to our participants, and thank you very much for your attention. Cheers.